dear students and viewers. Okay, today's the topic is DNA replication. Okay, so what is DNA replication? So அதை பற்றி நம்ம டீட்டெயிலாக பார்க்க போகிறோம். So already we talk about the central dogma of life. Okay, in the central dogma of life, the three major processes taking place: replication of DNA, transcription of DNA to RNA, and translation of RNA to protein. It's a two other process is called central dogma of life. Okay, the first step for the central dogma of life is the replication. Okay, of DNA. So they want to make a more number of copies by involvement of multiple enzymes. Okay, the major enzyme for DNA replication is DNA polymerase. Okay, so this class we are going to talk about the detailing what are the enzymes involving to make the more number of copies of DNA. Okay, that is for DNA replication. Okay, so first step, the replication. What will happening for the replication? The formation of new strand, okay, and sequence of nucleotides. The information is rewritten by the double stranded to the formation of new strands. Okay, during the DNA replication, each strand of exciting DNA molecule act as a template. Okay, this is the template. If you go for the single strand, formation of new strand from the old strand, the old strand they called as template. Okay, so synthesized to growing, okay, determined by the base complementarity. Already we told about ATGC. Okay, this is the base complementarity. Okay, so how this DNA replication mechanisms, that is for the semi-conservative mechanisms for the DNA replications. Okay, so the DNA replication is the process of formation of new DNA strand from the old DNA strand. The old DNA strand they called as template DNA. Okay, but the involvement of different enzymes. So the DNA replication in different options. So different options, if you look at the pictures number 1, 2 and 3. So most of the organisms, the DNA replication is the mechanism is semi-conservative mechanism. So you should know the difference between conservative, semi-conservative and dispersive. So semi-conservative means we get the off of the DNA from the old DNA to new DNA formation. But here the conservative, only one strand, the remaining strands were old strand, but dispersive, the DNA nucleotide dispersively they will present. Most of the living creatures and living organisms, the double helix DNA, from the double helix, the new strand DNA formation, the mechanism is semi-conservative mechanism. Okay, they will ask the question in final exams, even all the competitive exams, for which mechanism for DNA replication. They will ask semi-conservative, conservative, dispersive, or all the above. They will ask the question. The correct answer is semi-conservative mechanism. Okay. So how the semi-conservative mechanism, which scientists proved, yes, the DNA replication mechanism is semi-conservative mechanism. So Matthew Melson and Franklin Stagel in the year of 1950. So 1953 only they invented the DNA structure by Watson Crick, but the experiment, the cesium chloride equilibrium density gradient centrifuge methods, they will make the labeled DNA, the different labeled DNA with the cesium chloride chlorine solution with the bacterial DNA, they will label with the nitrogen, so that light DNA, hybrid DNA, heavy DNA. Okay, they did grown up, they will make the centrifuge based on the density gradient. They having the heavy, intermediate, lighter DNA. So they are getting the hybrid DNA, light DNA. Based on these experiments, they got this semi-conservative mechanism, the hybrid DNA. Okay, so they are identified with this experiment, cesium chloride equilibrium density ingredient centrifuge method. They Matthew and Franklin they proven all the DNA replication strategy through semi-conservative mechanism. Okay, so what are the enzymes involving to make the new DNA? Okay, replication of DNA occurs during the process of normal cell divisions. Okay, replication of DNA occurs during the normal cell division process because the genetic complement of the result daughter cells must same parent cell. Okay, while cell division, okay, from the parent cell to the results of the daughter cells, there should be a same the genetically fertility, there should be a same the rewritten the informations from the old cell to the new cell. Okay, the DNA replication must possess a very high degree of fertility. Okay, already I told about genetic fertility. Okay, genetically fertility means true to type, the resemble to the mother cells to the daughter cells, or true to type, the clones, the plant clones from the mother to daughters. Next generation should be uniform. But okay, so the entire process is a complex and involving multiple enzyme activities. So multiple enzymes activity. Okay, DNA replication to forming of new DNA molecules, they are involving multiple enzyme activities. They will ask the question, for DNA replication is a single enzyme to involving to make the new copy or multiple enzyme activities. They will question. Okay, so the major enzyme, one of the major enzyme is called DNA polymerase. Polymerase, to make more, poly means number. So the polymerase enzymes that synthesize DNA molecule from deoxyribonucleotides. Okay, so the polymer DNA polymerase to involving the DNA molecules making more number of copies. Okay, which scientists they identified or which scientists did the experiments and proven they got Nobel Prize for DNA polymerase. In 1956, Arthur Kornberg and Collicues discovered the DNA polymerase 1 in E. coli. Okay, so Arthur Kornberg is the scientist. He is the identified enzymes involving making new copies of DNA. So he is awarded in the for the for the field of physiology and medicine in the year of 1959. He got awarded this enzyme. They will otherwise called as Kornberg enzyme. Okay, they will ask question. What is Kornberg enzyme? The Kornberg enzyme is DNA polymerase 1. Okay, he is the scientist to identify to making new copy of DNA with help of DNA polymerase. The different kind of types of polymerase, the main role of this DNA polymerase, replicative and repair mechanism to make the new copies and repaired. So what is the DNA repairing mechanism? So we'll detailing we'll talk about the next slide. But before that, you should know the families and what are the polymerase present in the organism. Both eukaryotic and prokaryotic, they having polymerase 1 and polymerase Y. So different families, different examples. Okay, so you should know that making new DNA with help of DNA polymerase. Okay, so how the DNA replication is taking place, how the new DNA strand is forming, that is the very important. So if we for synthesizing of leading strand, synthesizing of lagging strands. So you know the 5 prime end, 3 prime end. Okay, the end of this DNA molecules we will call upstream end and downstream end. That's 5 prime end and 3 prime end. Okay, if you look at the pictures, see 5 prime to 3 prime, again 5 prime to 3 prime. The formation of new strand. The red color indicate is old strand. The blue color is indicating formation of new strand. Leading means continually the new DNA strand will forming. Lagging means discontinued. Okay, they will form the new DNA. They will called as Wakasaki fragments. Wakasaki fragments, otherwise called as lagging strand, discontinued strand. So how the DNA polymerase involving to make new copies, we'll see. But also the foremost important, the direction of replication, both prokaryotic and eukaryotics, is always 5 prime to 3 prime end. Okay, this is point by question. Which direction both the organisms, prokaryotic and higher organism, lower organism, with the direction the DNA replication is taking place. They will ask question 5' to 3'. Endonuclease and polymerase activities, exonuclease activities, they will go, they will bind inside and okay, forming of new DNA. So you know very well the backbone is sugar and phosphate. The inside of the sugar and phosphate nucleotide ATGC. This is the nitrogenous base pairs. So how it is forming? Okay. So multiple enzymes they are involving for activities to make new copies. There are the most 7 enzymes. Okay, 7 enzymes involving to make a new copy of DNA. Not only the DNA polymerase, not only for the DNA polymerase, but other enzymes also involving. Okay, so what are the enzymes involving to make the new copy of DNA? That is the big, if it is an important big question for final big question, and also for your UPSC exams, other competitive exams, they will ask the essay type question. What is the DNA replication? What are the scientists? How they identified, how they proved the DNA replication with the help of DNA polymerase? They will ask the essay type question. Even for all the competitive exams, what are the main enzymes involving DNA replication process? So if you take first enzyme, topoisomerase, single cell binding protein, primase, helicase, okay, RNA primer, DNA ligase, DNA polymerase, okay. So if you take 6 or 7 enzymes involved, there mostly the 7 enzymes to involve in take the new copies. Topoisomerase, okay, this is the parental DNA. The role of topoisomerase to binding to capturing the DNA. ஸோ எப்படி சொல்லணும், கயிறு திரிக்கும் போது கையை பிடிச்சிக்கிட்டு கயிறு திரிப்பாங்க தெரியுங்களா, அது மாதிரி தான் the topoisomerase to hold the DNA properly, to hold the DNA. So the helicase first to hold the DNA with the help of topoisomerase, then the helicase enzyme to unwind the DNA, to separate the DNA, okay, to separate the DNA, the primase also to make the nucleotides, then single cell binding proteins also the properly to separating the DNA. First என்ன பண்ணுவாங்க, ஃபார் எக்ஸாம்பிள் வந்து ஒரு த்ரெட் எடுத்துக்கிற, இல்லை ஒரு ரோப் எடுத்துக்கிற, ரோப்பை ரெண்டாக பிரிக்கிறோம். ஃபார் எக்ஸாம்பிள் உன்னுடைய தலையில் இருக்க சடையை எடுத்து ரெண்டாக பிரிக்கிறோம். ஸோ அது மாதிரி தான் ஹோல்ட் பண்ணிக்குவாங்க. ஸோ ரோப் மேக்கிங்லேயே என்ன பண்ணுவாங்க, ஹோல்ட் பண்ணிக்கிட்டு ஸோ த்ரெட்டை வந்து வைப்பாங்க. ஸோ அப்போ மேக்கிங் நியூ ரோல் வர மாதிரி தான் அந்த ரோலை நம்ம வந்து செப்பரேட் பண்ணுறோம். ஸோ ஃபர்ஸ்ட் topoisomerase to hold the DNA, the helicase activity to unwind the DNA, okay, to separate the helical strand, okay. Then the RNA primer they will involve to make the nucleotides. Both the side I told 5' to 3' direction, okay, 5' to 3' the DNA polymerase to repair and replicate and repair mechanism. See the both the strand and side they will make new strand with the help of major work is DNA polymerase repair and replication mechanism. So I told already lagging strand. See the lagging strand is discontinuous strand, okay, விட்டு discontinued இருக்கும். That is called as Okazaki fragment. The discontinued strand is called Okazaki fragment. This is one of the important question. The lagging strand or discontinuous strand, what is the called as Okazaki fragment. The discontinuous strand, the RNA primers and ligase molecules to fuse the DNA. But leading strand, they will continually to form new DNA. Old strand to new strand to form continuously. They call leading strand with the help of DNA polymerase. Here involving for DNA ligase to join the discontinued strand as well as to make the nucleotides, and also the polymerase involving repair and replicative mechanism. Finally they form new DNA strand, new helix strand from the template DNA, that's old DNA. So these are the overall view of DNA replication. So topoisomerase, primase, helicase, and DNA ligase, DNA polymerase to forming new strand of DNA. This is called as DNA replication mechanism. This is an important question. Okay, thank you.